Class, we're going to continue our discussion of respiration by going through the steps of glycolysis, which remember is going to make 4 ATPs. Now the formula for glucose is C6H12O6. We're going to follow what in our diagrams what happens to these 6 carbon atoms. But you need to remember that attached to those carbon atoms are also these hydrogens and oxygens. We're just not going to draw them in. Now what is glycolysis doing? It's preparing this big molecule of glucose for Krebs and etc. Where is it occurring? In the cytoplasm. Does it require oxygen? No. So what is it called? Anaerobic respiration. Now you learned in your chemistry unit that glucose, the 6-carbon glucose, is a monosaccharide. And it's the only carbohydrate that's small enough to enter the cell. Now, if glucose is barely small enough to get inside the cell, it's certainly too large to get inside the mitochondrion. So what glycolysis is preparing to do is to take this 6-carbon glucose and break it down into 2 smaller 3-carbon compounds called pyruvates for Krebs cycle. And it also forms some buses for etc. So, remember that glucose has carbon, hydrogen, and oxygen atoms, but we're only going to diagram the 6 carbon atoms. Now this is a little rough diagram of ATP, which is a molecule of adenosine with a tail of 3 phosphates, 2 of which are attached with high energy bonds. And if the cell breaks a high energy bond, it releases so much energy that a chemical reaction can occur. Now there's always some ATP present in the cell. So for glycolysis to occur, the cell has to spend a little bit of energy. This is like using a match to start a fire. So here are our 6 carbon atoms, and we've numbered them 1, 2, 3, 4, 5, 6. And what we're going to do is add phosphate to our glucose molecule. And whenever phosphate is added to a compound, this process is called phosphorylation. I put a star by that because that's a test question. Phosphorylation is the addition of phosphate to a compound. So, what we're going to do in the first step of glycolysis is one of these phosphate groups is removed from ATP and added to the number 6 carbon in glucose. This leaves a molecule of ADP and it forms glucose 6-phosphate. Now here is a diagram of glucose, galactose, and fructose. You'll remember all of these had the same formula, C6H12O6. They were all monosaccharides, and they were called isomers of each other. Isomers have the same chemical formula. They're made of the same atoms, but those atoms are arranged in different structures. So what happens next is that our glucose 6-phosphate is transformed into fructose 6-phosphate. The atoms are just rearranged. So the formula looks the same, but the atoms are arranged in a different pattern. So we go from glucose 6-phosphate to fructose 6-phosphate. Now the cell is going to spend another ATP. And we phosphorylate the fructose 6-phosphate. We add the phosphate to the number 1 carbon. We have ADP left over, and what's formed at the end of this is fructose 1,6-biphosphate. Next, we're going to cut this big compound in half, right here between the number 3 and the number 4 carbon atoms. And this forms 2 molecules of PGAL, or phosphoglyceraldehyde. Put a star by that because that's also some compound you need to know. So here's our diagram of fructose 1,6-biphosphate. An enzyme cuts it between the number 3 and 4 carbon atoms, and you get 2 PGALs formed. Now in step 5 of glycolysis, 2 chemical reactions occur at the same time, but we're going to diagram them separately. First, PGAL is phosphorylated, which, as we know, means we're going to add phosphate to PGAL. But the cell doesn't have to spend any more energy to do this. When we eat green and yellow vegetables, the phosphate that's in those vegetables is a mineral, and it's stored in the cytoplasm of the cell. So when we phosphorylate PGAL in glycolysis, the cell uses some of the phosphate that was in our diet that was stored in the cytoplasm. So here's PGAL being phosphorylated. Again, the phosphate is supplied by the cytoplasm, not by using an ATP. And you've got phosphorylated PGAL. Now PGAL has got 4 phosphates on it. How many ATPs are we going to make? 4. Here are the phosphates that are going to be used to make those 4 ATPs. Now the second chemical reaction that occurs in step 5, while phosphorylation is occurring, PGAL is oxidized. Now you know that when oxidation occurs, reduction follows it. Oxidation is a compound losing electrons, and reduction is a compound gaining those electrons. So what happens, and I don't know if y'all remember that oil rig, oxidation is losing, reduction is gaining. That's just a little way to remember what happens in an oxidation-reduction reaction. Well, PGAL is going to lose hydrogens. That's oxidation. At the same time that PGAL loses those hydrogens, NAD is going to pick them up. It's going to be reduced. It's going to accept those hydrogens. Now, NAD stands for nicotinamide adenine dinucleotide. Unfortunately, all of these processes have really big chemical names, but we abbreviated NAD, and one of the reasons why it's so important to have a good healthy diet is that the B vitamin niacin is required by our bodies to make NAD so that we can go through glycolysis in our bodies. Well, here's PGAL, and it's being oxidized. It's losing, what it loses is only one of the hydrogen protons and 2 electrons. We just diagram it H plus H. So it's losing hydrogen and NAD is gaining it. Losing hydrogen, NAD is gaining it. Oxidation, reduction. Oxidation, reduction. And what's left now is PGAL with 2 high-energy bonds. That oxidation-reduction reaction produces 2 high-energy bonds. And we've got NADH plus H buses, which are carrying that hydrogen to ETC. ETC stands for electron transport chain, and what it's transporting are these hydrogen electrons. Okay, if we go to step 6, now we're ready to make our first 2 molecules of ATP. We can make these ATPs by having an enzyme break these 2 high-energy bonds. The phosphates cut off the PGAL. It's added to ADP to make ATP. High-energy bond is broken. Phosphates added to ADP to make ATP. And when these high-energy bonds are broken, remember there's lots of energy stored in a high-energy bond, about 5 times more than what's stored in a regular covalent or ionic bond. So there's enough heat released to phosphorylate ADP. Now, we've got to make 2 more high-energy bonds. Remember, we've got 2 phosphates left, so we're going to make 2 more high-energy bonds. But this time, You remember from our chemistry unit, a dehydration reaction is also condensation, same thing, is the loss of water. So an enzyme comes in here and it cuts out a molecule of water from each one of the PGALs. And that's called metabolic water because it's, stays there in the cytoplasm and it's made by a chemical reaction. It's not something that you drank. And so what happens when this water is cut out of the PGAL molecule, a high energy bond is formed. Now we've got 2 more high energy bonds with a phosphate. So an enzyme comes in here, cuts that high energy bond, and the phosphate is donated to ADP, making ATP. That's what? Phosphorylation. So, for the test, not only do you need to kind of understand what's happening here in these steps, and I test you in order, what's occurring. But then you need to know the end results. And at the end of glycolysis, remember we want to make